And we'll continue our digestive system with the accessory organs, talking about the pancreas. So the pancreas is this thin leaf-like organ that is located posterior to the greater curvature of the stomach. And to orientate ourselves here, we have the stomach that is located more anteriorly in the upper left quadrant of the body. And the pancreas is located just posterior to that. And it has a head that is actually encircled by the duodenum or duodenum. And then the mid portion is called the body. And then the end on the left is the tail that's right next to the spleen. Well, the last time we talked about the pancreas, it had to do with the endocrine portion. So these were those pancreatic islets that would secrete insulin and glucagon and respond to blood sugar levels. Well, that endocrine portion is concentrated in the tail of the pancreas. The rest of the organ is going to function for exocrine, which is about 99% of the pancreas is exocrine function. So it's going to work to secrete digestive enzymes into the duodenum and release sodium bicarbonate. It's going to release about 1,200 to 1,500 milliliters of pancreatic juices a day. And because of the sodium bicarbonate, that gives this pancreatic juice an pH of less than 7. So it is considered to be basic. And that is beneficial because the chyme that gets released into the duodenum from the stomach is very acidic. So the sodium bicarbonate will neutralize the stomach acid. So that way the digestive enzymes that are going to be released in the intestines are not affected by the pH. And also, so some of the enzymes in the stomach, they might need to be activated. And so that can happen here in the duodenum in the presence of these pancreatic juices. There are also going to be acini cells that release their secretion into small ducts, and eventually those ducts will converge onto the main pancreatic duct. And so here we can see portions of the pancreas highlighted. We can see the head, the body, and the tail, and then we can also see the pancreatic duct going along the middle of that gland. Here we have some histology of that exocrine and endocrine pancreas. So this is just really showing how 99%, a majority of the gland is secreting exocrine. While we look over here at the endocrine pancreas, those are just those little islets. And so now we're at this portion. And so zooming in on the histology, we can see that the islets are these portions here. And so that is that that endocrine function. Whereas all of the cells around the islets, those are going to be the exocrine pancreas. And then we're also going to be able to see some other structures like arteries and veins and ducts. So we can notice that the veins here highlighted, and then we've got the arteries. If we zoom into the microscopic anatomy of the pancreas, we can see that there's those ducts, and they're lined with those acinar cells. So we have a simple cuboidal epithelium that's going to line that duct. Well, that duct is going to lead to another duct that's going to eventually lead to a bigger duct. And those ducts are going to get bigger and bigger until eventually they lead to that main pancreatic duct. So here we have parts of the histology highlighted. So we have the acineus, which are those cells of the pancreas. And then we have the duct of the pancreas with that simple cuboidal epithelial tissue here and here. And so eventually these ducts are all going to converge and lead to the main pancreatic duct. And that pancreatic duct is going to run along the middle of the pancreas. Well, eventually it's going to lead to the duodenum, but before, right in the head, it's going to actually converge and join up with the bile duct coming from the liver. And what's going to allow this pancreatic juice and the bile coming from the liver is the hepatopancreatic sphincter. So that will open and close to allow these gastric or the bile and the pancreatic juice into the duodenum. There is also another duct that is a little smaller. It's called the accessory pancreatic duct, and that branches from the main pancreatic duct. And I have a picture coming up next to show you. But that will actually kind of bypass, it's like a detour, and it goes to the duodenum independently. And when it does this, it actually bypasses that sphincter to allow pancreatic juice to be released into the duodenum even when bile is not being released. And so here we have that image where we've got the leaf-like pancreas, but the stomach's now been removed. You can see the duodenum wrapping around the head of the pancreas. And then the pancreas running along the middle will have that pancreatic duct. And that duct will meet up with the common hepatic duct from the liver, and it will form that hepatopancreatic ampulla. And there's going to be that smooth muscle forming the sphincter, and then you'll see the major duodenal papilla that projects into the duodenum. And then up here we have that accessory pancreatic duct that bypasses that sphincter to allow the pancreatic juice to be released, even though bile will not be. And so these pancreatic juices that are released, it is a watery, basic, or alkaline mixture. And what makes it basic is that sodium bicarbonate that's inside of it, some electrolytes, enzymes, and zymogens. So recall that zymogens are proenzymes. These are inactive precursors of an enzyme. So they're like the before. They're not turned on yet. They require something to activate it. So we have acini cells that will secrete enzymes and zymogens within the pancreas, and those will go down into the ducts, and those ducts will eventually lead to the pancreatic duct, and that pancreatic duct will go into the duodenum. And so there in the duodenum, there might be something like enteropeptidase that is located on the brush border of the duodenum that can activate some of these zymogens. And those ducts will also be releasing that sodium bicarbonate, which will function to neutralize the stomach acid because that chyme has hydrochloric acid in it, and that would literally burn a hole through the intestines. So we got to neutralize that acid, which is where the sodium bicarbonate comes into play. So the pancreatic zymogens are trypsinogen, chymotrypsinogen, and procarboxypeptidase. And so when we're talking about these zymogens, anytime you want to figure out what it's going to become, you just take out the OGEN at the end. And then whatever's left, the beginning, that is what it will become. So trypsinogen will become trypsin. And so trypsinogen is secreted into the intestinal lumen. It is going to then be converted into trypsin by the enterokinase that's secreted by the small intestinal cells. Trypsin will then be what converts chymotrypsinogen to chymotrypsin, and it will also convert procarboxyl peptidase into carboxypeptidase. And so those three zymogens are activated within the small intestines, and they're all involved in protein digestion, meaning they'll take those proteins and they'll break them down into their building block or monomer called an amino acid. Diagram is showing the pancreas and how those zymogens are released into the duodenum in inactive form, and then they're going to be converted to become activated. So trypsinogen is released into the duodenum of the small intestines, and that intestines is lined with intestinal cells that make enteropeptidase. Don't be afraid of that big scary word. Entero just means that enteric tract, your alimentary canal. Peptide means protein, and ACE means that it's an enzyme that's going to break that substance down. So this is an enzyme that breaks down proteins in the intestines. So that is going to turn or convert trypsinogen into trypsin. And now trypsin can convert the other two. So trypsin will convert the chymotrypsinogen to chymotrypsin and the procarboxyl peptidase into the carboxyl peptidase. There are some other enzymes that are also secreted by the pancreas, like pancreatic amylase. Just like salivary amylase that's released in the mouth, it digests carbohydrates. Pancreatic amylase will digest starch. So starch will be broken down into its monomer, or its building block of a monosaccharide that we call glucose. And then there is pancreatic lipase that digests fats. So that will break down fats into their monomers or building blocks of glycerol and fatty acids. And then there's one we don't talk about as much called ribonuclease and deoxyribonuclease. These are enzymes that are going to break down nucleic acids like DNA and RNA into their nucleotides. And so it's going to be something that tells the pancreas to start secreting these substances. It'll tell the pancreas what to secrete and when to secrete. And this is going to be similar to what tells the liver and the stomach to secrete. So part is the parasympathetic nervous system, where we have the vagus nerve that's going to release acetylcholine. And then we will also have acetylcholine being released from those intestinal nerves as part of the enteric nervous system, specifically during the cephalic phase of digestion. And this acetylcholine stimulates the acini cells to secrete enzymes even before food is swallowed. And the enzymes will remain in the acini cells and in the ducts until the chyme enters into the duodenum. Cholecystokinin, as we abbreviate it as CCK, that is stimulated within the duodenum of the intestines. And so once fat arrives within the small intestines, CCK is going to start to get released by the mucosa. And that is going to then have an effect on the acini cells in the pancreas. So it's going to have an effect on that neighboring organ. And that will tell those acini cells to release those enzymes. At the same time, the gallbladder is also going to respond to the CCK, and it's going to stimulate that gallbladder to start contracting to release the bile that is stored so fat can start being broken down. And then the CCK also is going to have an effect on that hepatopancreatic sphincter. Because if the sphincter is closed, then there's no good in releasing the secretions. So we got to open up that sphincter. So the hepatopancreatic sphincter will open, and that way the bile can be released into the duodenum as well as the enzymes from the pancreas. The third regulation of secretion is called secretin. And this hormone comes from the cells within the duodenum of the small intestines. So once that acidic chyme arrives there, these cells are going to say, whoa, we got to neutralize that. So secretin is going to be released, and that will stimulate the pancreas to start to release some more sodium bicarbonate. It's also going to raise the pH level that is required for some of these pancreatic enzymes and intestinal digestive enzymes. Some of them won't work or they'll be harmed by a low pH. So the sodium bicarbonate raising the pH level is going to allow these enzymes to start working. The secretin will also have an effect on the liver, having the liver begin to produce more bile. And then the ducts of both the liver and the pancreas will also be stimulated to allow that bile and the sodium bicarbonate to be released. This diagram shows you stimulation by the vagal nerve that's going to cause the pancreas to release those pancreatic juices, and it's also going to stimulate the gallbladder to contract. So you can see that nerve here, and it's going to branch off and innervate the gallbladder at the same time as this pancreas. So once we have this chyme entering into the duodenum, that's going to cause these enteroendocrine cells of the small intestines to release secretin and CCK. CCK and secretin both enter into the bloodstream, and they're going to be able to travel locally to that pancreas and locally to the liver. Well, in the pancreas, CCK will induce will induce secretion of enzyme-rich pancreatic juices. Secretin then causes the sodium bicarbonate to be released. Secretin is also going to impact the liver to make more bile. So the gallbladder will be stimulated to release its stored bile, and the liver is going to start making some more bile that can then be immediately released, and then any extra will be stored.